Well, thank you, Robert, and thank you all very much for turning out in this interesting time. And it's a real privilege to be here at Cornell. I've never been here before, and it is the most beautiful place. Ithaca is the most beautiful town, and I'm really enjoying being here. So thank you for turning out tonight. This, I must warn you, is quite a technical presentation. Most of my presentations are basically telling stories, and this is not like that at all. So it's kind of technical, but it's in language, hopefully, that everybody will understand. I'm more than happy to take questions at breaks during the presentation or at the end, hopefully both. So tonight, I'm going to be asking the question, is Cornell misleading us on GMO safety? These are two resources that if you want to know more, I recommend you go to and look up. The free download of GMO Myths and Truths is available online on www.earthopensource.org. The updated version, GMO Myths and Truths, the book, is available from Amazon. This is the shorter one. It's therefore probably more reader-friendly, and I do recommend that you try it out and read it. So, GMO safety debate is over. So says the Cornell Alliance for Science. In May 2016, Cornell's Alliance for Science published an article by the British GMO promoter Mark Linus. He's not a scientist, he's an environmental writer. He specializes in climate change. And this article proclaimed that the GMO safety debate is over and that people need to stop being scared of GM foods. And Linus focused on a report by the US National Academy of Sciences, or NAS, which found, and I quote, no substantiated evidence that foods from GE crops were less safe than foods from non-GE crops. However, Mark Linus is a non-scientist. He's paid by Cornell's Alliance for Science to promote GMO crops to the exclusion of almost everything else. Those are Mark Linus's words. The Alliance for Science is funded by the Gates Foundation. I've already seen the building here that Gates founded. And Gates has invested heavily in Monsanto and the agribiz giant Cargill. The National Academy of Sciences, the NAS, and its research arm, the National Research Council, have actually taken millions of dollars in donations from Monsanto and other biotech companies. And no less than 11 of the 19 experts listed in the NAS report on GM actually have ties with the GMO industry or with GMO advocacy. Now, the truth is more likely like this. This is a paper that was published in a scientific journal. It has the title, as you can see, No Scientific Consensus on GMO Safety. It was drafted and written by a group of very eminent qualified scientists on this issue, and signing up to this statement on the European Network of Scientists for Social and Environmental Responsibility website, there are over 300 qualified scientists who have actually signed up to this statement to support it. This is a peer-reviewed paper. As you can see, the title is An Illusory Consensus Behind the GMO Health Assessment. And this review of the scientific literature concludes that the claimed GMO safety consensus is illusory and that it's influenced by politics and corporate interests. This is from a peer-reviewed paper published in a journal. It's about the common toxic effects of GM foods as revealed by animal feeding studies. And this is a quote from that paper. It says, the results of most studies with GM foods indicate that they may cause some common toxic effects, such as hepatic, that means liver, pancreatic, renal, that means kidney, or reproductive effects, and may alter the hematological, that means blood, biochemical, and immunologic parameters. That's a pretty strong statement. So what is a GMO? You guys will probably know this already, but we'll just recap. A genetically modified organism, or GMO, is defined by the World Health Organization as an organism in which the genetic material, or DNA, has been altered in a way that does not occur naturally. How is genetic engineering done? It involves manipulating the organism's genetic material, or genome, in the lab by forcibly inserting pieces of DNA from other organisms or by modifying, editing the existing genetic code. It reprograms the cells of the GMO to make a new protein or to modify an existing protein, and it confers new properties or traits that are not naturally present in the organism. Should we worry about GMO food safety? Well, GMO crops are produced through a procedure that's radically different from conventional plant breeding. And this is not a message that you will hear much in America. It's not a message that you'll hear from the mainstream media. However, this is the scientific truth. It does not involve sexual reproduction. It allows the transfer of one or a few genes between totally unrelated organisms using artificial combinations of genetic material. It allows the crossing of species barriers to reproduction in ways that do not occur naturally. And it produces combinations of genes that have not evolved to work together in a coordinated, integrated whole. And the GMO transformation process as a whole, that means the transgene insertion plus the tissue culture that they use to reproduce the plants, is highly mutagenic. And mutagenic simply means that it damages DNA. So why should we worry about GMO food safety? The combined effects of GM, that is mutation effects and the novel combinations of gene products, can disrupt genetic and protein biochemical function in the plant. They can lead to the generation of novel toxic effects, in particular allergies and altered nutritional value, and also new toxins that weren't there before. So GM crops pose new risks to food safety that need to be evaluated for acute and especially chronic, that means long-term, toxic effects. Now, the commercial GM crops that are out there right now are modified to tolerate herbicides or to produce insecticides. 58% are herbicide tolerant, mostly to glyphosate herbicides, that's Roundup to you and me. They absorb herbicide residues without dying. So the farmer can spray the crop with a ton of herbicide and the crop will survive, but all the other life, all the weeds in the field will die. At least that's the hope. In reality, of course, what's happening is that the weeds are getting resistant to the herbicide and they are surviving. So there is soy, corn, cotton, canola and sugar beet that are now herbicide tolerant and can be sprayed with massive amounts of herbicides. 15% of commercialized GM crops are engineered to produce a modified insecticide, for example, BT corn and BT cotton. 27% both absorb and produce pesticides, and they combine both traits, for example, Smart Stacks corn. Now, the other side will tell you, the pro-GMO crowd will tell you that GM crops are being grown all over the world by farmers and there's really no problem, because if there was an issue with GM crops, farmers wouldn't be growing them. That isn't true. 90% of GM cropping is just in 6 nations. The US leads, and following the US is Canada, Argentina, Brazil, India, and China. Claims about the need for GM crops. We're told that they create more sustainable agriculture by reducing chemical inputs, pesticides and so forth, that they're easier for farmers, that they will feed the world. This is one that we hear all the time. Most articles in the newspapers and also scientific papers will start with this claim that GM crops are needed to feed the world. In order to do that, it is implied that they have higher yields. We're told that they will create more nutritious foods and that they'll help us meet the challenges of climate change. Unfortunately, what the claims of need for GM foods ignore is that we already produce enough food to feed 14 billion people, and that's double the world's population and more than we will ever need. In 2050, the predictions are that the population will level off and then possibly start to decline. So we already produce enough food. There is no shortage of food, quite the reverse. We feed enough grain to animals to feed 3 billion people. In the industrialized world, 40 to 50% of all food is wasted. And there are large losses of food in the developing countries due to a lack of infrastructure, for example, storage facilities. There's also large diversion of food crops, including soy, maize, and sugarcane, to biofuels. And biofuels are really a way of using up agricultural surpluses. That's what they're about. In addition, there are no improvements in crop performance from GMOs. There's no intrinsic yield increase. In fact, with soy, the GM soy actually has a yield drag. It yields worse than non-GM soy. Pesticide use has also increased overall with GM crops introduction. There is a better way. Agroecology is the key to future food security, and that's not just me saying it. There's good peer-reviewed data. This is a report called the IASTED report on the future of farming. It was authored by 400 international experts, scientists and all kinds of experts, agronomists and so forth, published in 2008, and it found that low-input agroecological farming practices have proven to be sustainable and resilient, that they provide higher yields, diverse nutritious diet, and can ensure food security. This report also concluded that GM was not needed to feed the world, and the authors said that the yields were variable for GM crops, safety questions remain, and intellectual property rights, in other words, patents on the GM crops, are a concern, particularly to the developing world. This is one that you'll hear everywhere. The Americans have eaten trillions of GM meals and they're absolutely fine. Well, apart from the fact that we know that Americans are not fine because there are some pretty serious health problems here, there are other issues with this argument as well. This argument is very unscientific and dishonest because there have been no human population epidemiological studies to show the effects of eating GM crops. Nobody knows. Such studies are not possible on the continent where most GM meals are consumed, North America, as GM foods are not labelled. You've just been subjected to a strange act that has been passed in this country, which we call the Dark Act, which is supposed to tell Americans whether their foods are genetically engineered or not. Actually, the information is hidden behind things like barcodes, and you'll need a smartphone and an internet connection, and you'll need to be pretty technologically savvy in order to find out whether it contains GMO ingredients. So this is really a travesty of a public information law. Now, there is also a study that you may have heard of, which is claimed to say, this study, that 100 billion animals that have eaten GM feed show that GM is safe. This is a study led by a former Monsanto scientist called Alison van Eenennaam, and it claims that data from over 100 billion farm animals collected over 29 years, both before and after the introduction of GM feed in 1996, show that GM crops are safe. However, this study actually says very little about GMO safety. It's uncontrolled for many variables, including the escalating antibiotic use in livestock, and that can mask health problems. It hides inflammation and other diseases in animals. There's no information in the study on how many of these animals were eating GM feed, how much, and for how long. An astonishing 98% of these 100 billion animals are poultry, and 92% of the 100 billion animals are 49-day-old broiler chickens. And these chickens are completely irrelevant models for assessing human or even mammalian health risks. It tells us nothing about those things. 49 days in a broiler chicken is actually a small fraction of a chicken's natural lifespan, which is 5 to 7 years. So in fact, this study gives no long-term data even in chickens. There's no detailed health data on the animals. The only health examinations seem to be slaughterhouse inspections, which are sporadic and they miss illness in animals. For example, a study by Dr. Judy Carman found that GM feed caused more severe stomach inflammation and heavier uteri in pigs. We'll come back to this study a little later. However, all the animals in this study passed the slaughterhouse inspections as completely safe, and they were able to enter the food supply. So you can have a sick animal, it can still be passed as perfectly fine for the food supply. The NAS, the National Academy of Sciences, has, however, used this study to claim GMO safety, in spite of its evident weakness. So the NAS's elevation of the study actually led a veterinarian and a critic of badly designed animal feeding studies with GMOs, Dr. Ina Valikov, to accuse the Academy of Sciences of attempting to turn chicken shit into diamonds. This is another misleading review of animal feeding studies with GMOs. It's by Snell and colleagues, published in 2002. It claims to be a review of 24 long-term animal feeding trials with GM crops. It concludes that the GM foods tested are safe, and this review is also used by the US National Academy of Sciences as evidence of GMO safety. Problems with the Snell review are that some of the studies in this review are not long-term at all. So you have chicken studies of 35 to 42 days. Chicken's natural lifespan is 5 to 7 years, so it's just a fraction of their lifetime. There is a dairy cow study of 25 months. Again, this is not long-term because the natural lifespan of a cow is 17 to 20 years. So these studies give no information at all on long-term health effects. There are also double standards used in the Snell review. This is very unscientific. Snell dismisses all findings of ill health effects in the animal feeding studies with GMOs based on weaknesses in the study methodology. However, the Snell review accepts at face value findings of safety for GM crops, even though the studies that conclude that the GMO tested is safe suffer from exactly the same weaknesses. So this is asymmetrical, unfair application of double standards to scientific studies. And it's an absolute travesty that studies like this are being published in the peer-reviewed literature. You wonder what's happened to science. Snell and colleagues actually cite a study in dairy cows. This is a study by Steinke et al., a German study published in 2010 as showing GMO safety. However, half the cows in the test and the control groups were removed at some point during the study due to sickness and infertility and replaced with new cows. And this completely invalidates the conclusion of the study that no GMO-related effects were seen. Clearly, it's never acceptable to replace the experimental subjects halfway through a study. It's something that is simply never done, except in these really strange tests with GMOs that for some reason are peer-reviewed and passed and published. It's almost unbelievable. There is a shortage of long-term studies on GMO foods. This is worrying because we know by now that GM foods are very unlikely to be acutely toxic. They're not the kinds of things that you would necessarily eat and immediately get ill and die. We suspect that is not going to happen. It's the long-term effects that are of concern. And it's exactly the same with things like tobacco. You don't smoke a cigarette and then drop down dead. It takes time. So the shortage of long-term studies on GMO foods was emphasized by the French Food Safety Agency, ANSES, in its critique of the Seralini study on a herbicide-tolerant GMO maize and the Roundup herbicide that it's grown with. This was a very controversial study. You may have heard about it. You may have heard it was a rubbish study. That actually is not true. It was a limited study, as all scientific studies are. They have their limitations and weaknesses, but it was not a bad study. So ANSES searched for long-term animal studies on herbicide-tolerant GMOs and their associated pesticides to compare with Seralini's study. ANSES found only 2. And these herbicide-tolerant crops, remember, constitute over 80% of all the GMOs that are in the food supply in the world. So it's a pretty major thing, and ANSES could only find 2 studies to compare with Seralini's. One of these studies by Professor Malatesta's group in Italy actually found health problems, including more acute signs of aging in the liver in mice fed GM glyphosate-tolerant soybeans compared with the controls. The other one was a Japanese study which found no meaningful difference between rats fed GMO glyphosate-tolerant soybeans and non-GMO soybeans. However, the glyphosate in the GMO soy was measured, and it was only found at the limit of detection. So that suggests that the GM soy was sprayed only once, perhaps early in the growing season, or maybe even not at all. And this does not reflect the real-life way in which GMO soy is grown. We'd expect it to be sprayed at least twice with Roundup, and probably other chemicals as well. The regulation of GM foods. Are the regulators protecting us? Well, not really. The regulatory standards are very highly variable around the world, and everywhere you go, they are inadequate. I come from Europe, which is supposed to have the best GMO regulatory system in the world, and it's extremely weak. The US system is much weaker, but wherever you go, it's incapable of finding risks from GM foods. One problem is that the regulators never question the technology in the context of the current understanding of genetics. And I believe that you've already had lectures in this series about that, probably from Jonathan Latham, just explaining why the GM technology is based on very outdated idea of how genes work. Another problem is that regulators only accept data from the GMO developer company. And when I tell the public this, there's normally a sort of shock response among some people, because somehow the public expect that the regulators will be doing their own tests on the GMO foods, or they'll be commissioning independent scientists to do tests on them. Well, that doesn't happen. All the data comes from the company that wants to commercialize this GM crop. So that's a problem of bias. There are major flaws in the industry study designs. For example, they include spurious, non-genetically equivalent comparators for the GM crop and historical control data. Now, those are quite technical terms, but really, I don't have much time to explain those in detail tonight, but all you need to know is that those 2 practices actually introduce a lot of noise into the data set, so that you then cannot see any effects that are due to the GM crop, because there's so much noise in the data set that no difference is apparent. They do a superficial compositional analysis, or rather the company does it, in order to conclude that the GM crop is substantially equivalent to the non-GMO parent. And basically this consists of measuring some basic components in the GMO crop like carbohydrate, protein, and fat. And if it's roughly within the range of what you find in the non-GMO parent, they say, well, it's substantially equivalent, and therefore we think it presents no particular risk. There is also no requirement whatsoever anywhere in the world for long-term toxicity studies in animals. What are the sources of toxicity for a GM crop? There are 3, at least that we can think of. One is the intended gene product. For example, the Bt insecticidal protein in GM Bt crops people who eat BT crops are eating an insecticide, and there are plenty of reasons to think that that might be a problem. It could be unintended changes in crop biochemistry as a result of the disruption of the genes during the genetic engineering process, which is very disruptive to the DNA. So it can change the biochemical pathways within the plant. And the result of that can be things like enhanced toxins in the plant, or enhanced allergens, or new toxins and allergens. There are also herbicide residues in GM herbicide-tolerant crops, for example, glyphosate, and now herbicides like 2,4-D and dicamba, because the industry has now developed GM crops that tolerate other herbicides as well as Roundup, in particular 2,4-D and dicamba. And these are very highly toxic herbicides. Nobody thinks almost that they are fine and okay, except the regulators, of course. So 80% of GMO crops are engineered to tolerate glyphosate herbicides, for example, Roundup, so they will contain herbicide residues, and we will be eating those along with the food. Controlled animal feeding studies show clear signs of toxicity from GM crops. This involves both Bt toxin and glyphosate-tolerant GM crops like soy and corn. We found that they can lead to damage to multiple organs and systems, both in terms of structure and in function. For example, the liver and the kidney. These are two commonest organs that are affected in these animal feeding studies. And also problems with the immune system, allergic type reactions and immune responses. And that, to me, is an issue, because I see the rising allergies and all kinds of immune problems, especially in children, and they are really escalating. So we should be looking at GM crops to see if there's a connection. Also, problems in the digestive system. We see gut lining cell proliferation. That can be a prelude to cancer. It may not be, but it can be. And also lesions in the gastrointestinal system, things like ulcers and sores. So breaking down now into the individual studies, this is just a series of data points. I apologize that in some ways it's not very interesting, but it's basically to show you that there is hard data behind what I'm saying. So these are studies by academic scientists in universities and public institutes. Rats fed GM-BT corn over 3 generations suffered necrosis in the liver and kidneys and alterations in blood biochemistry. Necrosis simply means areas of dead tissue. Old and young mice fed GM-BT corn suffered marked disturbance in immune system cells and in biochemical cytokine activity. Cytokines are proteins that are involved in the immune system. Pigs fed BT corn for 31 days suffered differences in immune cell type numbers and also in biochemistry, again, cytokine levels. Ewes and their lambs fed GM-BT corn over 3 generations. The ewes showed excessive growth of the cells in the lining of the rumen. The rumen is a bit of the gastrointestinal tract that sheep have and humans do not. The proliferation of cells indicates that the GM maize caused damage to the rumen lining, and the authors suggest that this could be due to the Bt toxin. And also disturbed gene functioning of liver and pancreas was seen in the lambs. Rats fed GM-BT corn for 91 days, suffered multiple organ changes in weight and biochemistry. There was damage in the structure and the function of the liver, kidney, testes, and intestines. So multiple organ problems there. This was a long-term toxicity study on pigs fed a combined GM soy and corn diet, and it was by Dr. Judy Carman, who's an Australian scientist, and she collaborated with a farmer in the US to do this study. And she found that there were gastric and uterine differences in the GM-fed pigs. There was a marked increase in severe stomach inflammation, 4-fold in males, 2.2-fold in females, and the uteri were 25% heavier. And you see these pictures here, starting clockwise from top left. That is the stomach of a non-GM fed pig, and it has no inflammation. The next one to the right is a stomach of a non-GM fed pig, and you can see very mild inflammation. It's just very slightly pink. On the bottom, on the right, you see a stomach C34. That is a GM fed pig. It has moderate inflammation. You see that the colour is darker pink. And to the left of that is another stomach of a GM-fed pig with severe inflammation. So quite marked differences. Industry studies on rats fed BT corn suffered signs of toxic effects to liver and kidney function. So there's a number of studies showing that. Some French scientists did a reanalysis of feeding studies conducted by industry on a BT corn, and they found that the industry data, this is Monsanto data, showed that the GM-fed rats grew more slowly, they had higher levels of certain fats, triglycerides in their blood, and they had problems with liver and kidney function. And in the chart on the right there, you don't certainly need to read that whole chart, but just take note of the little boxes that are drawn around the figures. Each one of those boxes represents a statistically significant difference in the GM-fed animals. So you see, there are a number of biological differences in animals that eat GM feed. This is a reanalysis by Seralini's group, again in France, reanalyzing Monsanto's data from feeding studies with GM-BT corn. Again, signs of toxic effects on the liver and kidneys. Again, look at that chart, look at the boxes, and you will see that those boxes contain statistically significant differences in the GM-fed animals. Now, this is Monsanto's response to that reanalysis of their data by the French team led by Seralini. And it's very small, so I'll read this out to you. But this is the highlighted bit from the abstract. Monsanto says that statistical findings reported by both Monsanto and Seralini were considered to be unrelated to the treatment. That means they were nothing to do with the GM. Or they were of no biological or clinical importance because they failed to demonstrate a dose-response relationship, reproducibility over time, association with other relevant changes, for example, histopathology, occurrence in both sexes, difference outside the normal range of variation, or biological plausibility with respect to cause and effect. The Seralini reanalysis does not advance any new scientific data to indicate that MON 863 GM corn caused adverse effects in the 90-day rat study. So Monsanto's coming up with all these excuses of why these statistically significant differences are not biologically relevant. And all these points are actually not scientific. This is trying to reinvent science. For example, Monsanto is claiming that you have to have the same effect in the male and the female rats before it can be taken seriously. Now, all of us should know that that is complete nonsense. Males and females have different hormonal systems, and they can have different health effects from a particular agent that you're testing on them. Also, the one that I like best or dislike most is where Monsanto says that it lacks biological plausibility, as if Monsanto has to believe that there's something wrong with the GM corn in order for there to be something wrong with the GM corn. In other words, if Monsanto doesn't believe it, then it doesn't exist. This is not science, it's propaganda. So this is, again, another industry study, rats fed commercialized Roundup tolerant this time, GM NK603 corn for 90 days. Again, signs of toxic effects on liver and kidney. Academic studies, mice-fed commercialized Roundup-tolerant GM soy. The mice-fed GM soy suffered disturbed liver, pancreas, and testes function, and also more acute signs of aging in the liver. Are these effects caused by the GM process or the Roundup residues or both? We still don't know. There have been 20 years of animal feeding studies with GMOs, and we still don't know in most cases whether the effects are due to the herbicide residues in the GM food or something to do with the GM food in itself. And this is a fundamental failing of scientific research into GM. The scientific climate now is so stifling that scientists fear doing research on these crops. They don't want to find anything wrong with these GM crops because they're frightened. They're frightened of losing their jobs. The Italian scientist who did these studies, Professor Malatesta, she wanted to follow up her studies afterwards to find out whether the ill effects in the GM-fed rodents were actually due to the Roundup residues in the GM soy or something to do with the GM crop itself. She tried to get funding and she couldn't get funding. And what she was told was nobody wants to fund these studies because everyone is frightened of Monsanto. So we do know that the GM process itself can cause toxic effects, and this is just one study that illustrates that. It is an industry study on GM tomatoes, and these were, if not the first GM commercialized food, certainly one of the first. So these rats fed GM tomatoes over 28 days developed stomach lesions, sores or ulcers. There was unexplained high mortality, death rates in the GM-fed rats. 7 out of the 40 rats fed the GM tomatoes died within 2 weeks of the start of the experiment. And in the non-GM-fed groups, they didn't have any deaths. Evidence of mutagenic effects of the GM transformation. This could be what is explaining these problems with GM foods. Use of molecular profiling technology has shown, in the case of commercialized BT corn, it's found marked alteration in protein composition profile specifically related to the GM transgene insertion event. Significant differences in virtually all types of biochemical components and alterations in amino acid profiles. So all these things can affect toxicity, they can affect allergenicity, they can affect nutrient value. Non-commercialized low glycoalkaloid GM potatoes, when they analyzed these, they found unexpected significant changes in several other metabolites, decrease in chlorogenic and caffeic acids, glucose and fructose. All long words. I'm not altogether sure what they all mean. However, I do know that all those substances are biologically active and they can make a difference to our health. So evidence of mutagenic effects of GM transformation continued. Here are some more. Non-commercialized GM rice. This was a GM rice engineered to be resistant to fungal diseases, and it had markedly altered seed structure and composition compared with the non-GM parent. GM rice engineered with Bt toxin and other insecticidal genes marked biochemical and nutritional disturbances, again compared with the non-GM parent. So you see, GM can produce some unexpected, unintended changes in the GM crop, and those can have biological significance. After molecular profiling, it's extremely unlikely that any GM crop could be claimed to be substantially equivalent to the isogenic non-GM variety. Isogenic simply means genetically the same. That means the non-GM parent. So feeding studies conducted by academics on non-commercialized GM crops. This was a study on GM potatoes. These GM potatoes were supposed to be commercialized. This was the famous Arpad Pushtai study, which was conducted in Scotland in the UK. The rats were fed GM potatoes for only 10 days. This was an incredibly short period of time. Even so, they showed excessive growth of the lining of the gut, similar to a precancerous condition, as well as toxic effects in multiple organ systems. The pictures that you see on the left, that's the GM-fed rats. You see that the lining of the gut is much thicker. It's increased, and certainly compared with the non-GM. And that can be signs of a precancerous condition. So rats fed GM-BT rice, significant differences in gut bacterial populations and organ weights, including adrenals, testis, and uterus. Study on GM peas caused a surprise allergic reaction. There were unexpected changes when these proteins were engineered into peas, and it ended up that the peas caused a marked immune response and allergic type reactions in mice. So again, that's pretty serious, that all of a sudden a food can become allergenic to some subjects when it wasn't before. There are some pretty serious scientific insufficiencies in the health risk assessment of these 19 commercialized agricultural GM crops. The 90-day toxicity tests of these crops revealed signs of liver and kidney pathologies. Again, these have all been commercialized. They're all in the food supply. So to summarize what we've just seen, studies on GM commercialized soy and corn show consistent signs of toxic effects in the liver and kidney structure and the function, as well as immune system disturbances and gut lesions. And these effects have been seen mostly in short and medium-term studies, but they may be markers of the onset of chronic disease. So long-term studies are needed to assess this more thoroughly. But unfortunately, long-term studies on GM foods are not required by regulators anywhere in the world. In the EU, at present, we have a requirement for a 90-day rat study, and the regulators are trying to do away with that. So what could be causing these signs of toxicity? Just to recap, it might be the introduced foreign GM gene, transgene. It could be the GM gene product directly, for example, Bt toxin. It could be the altered plant biochemistry caused by the GM gene product, for example, enzymes conferring the herbicide tolerance. It might be altered plant biochemistry caused by the mutagenic effect of the GM transformation process. Again, mutagenic just means DNA damaging. And it might be higher exposures to herbicides used in conjunction with the GM crop, for example, the glyphosate formulations. Glyphosate is a problem. As we've mentioned, 80% of GM crops are engineered to be tolerant to glyphosate. It's now the world's most used pesticide, 100-fold increased use since 1974. Two-thirds of that total use has been in the last 10 years. So you can see that use is really escalating quite steeply. The majority of GM soy is herbicide tolerant, Roundup tolerant. And GM soy has been found to contain levels of glyphosate that Monsanto itself has called extreme. Here's some examples of average levels. These are not the highest levels by any means, but they are significant amounts that we're ingesting. Industry and regulators say this doesn't matter because glyphosate Roundup is safe. You see these people are spraying it merrily in their gardens and on verges without any protective equipment at all. And that, I'm sure, is the norm. This guy knows better. He's wearing a special suit to protect him from the effects of the Roundup. He's putting it into his canister for crop spraying from the air. He's actually a pilot that's going to spray this stuff from the air. Glyphosate is claimed to be safe by industry and regulators due to its perceived low toxicity, and that's based on the fact that its weed-killing ability works on the basis of its inhibition of a certain enzyme. Now, this enzyme is present in plants and some bacteria and fungi, but it's not present in humans or other animals. And so industry's claim has always been that glyphosate does not affect humans, it doesn't affect mammals, because we don't have this pathway. That is a problem assumption, because there are other pathways through which glyphosate herbicides can exercise toxicity, as scientists have found. They also claim that there is rapid excretion via urine and feces. In other words, you pee out the glyphosate that gets into you. They say that there's no bioaccumulation. However, that has not been tested scientifically in a way that is representative of the kind of exposures that you and I will have. So it is an assumption. These are the current acceptable daily intakes for glyphosate that are set around the world. In America, you call it a chronic reference dose. You can see the first thing that you'll notice is that they're very different. So in the EU, we have a much lower allowed level of glyphosate. China and Russia is a little bit higher, and you see the highest one we've ever found is in the USA. So the US regulators think it's a lot more safe than the EU regulators think. Are these exposures really safe? Well, I got together with a group of scientists some years ago, and we analysed the industry studies on glyphosate, and we looked in particular at those animal feeding studies that were looking at birth defects in the animals fed glyphosate. What we found is that based on the industry studies alone, the acceptable daily intake for Europe should have been set at least 3 times lower than it currently is. If you take independent science into consideration and not just the industry science, the level should be at least 12 times lower. So we think that these acceptable daily intakes are really not scientifically based. Glyphosate has multiple toxic effects. You may have heard about some of these. They have been in the news recently. It is an antibiotic. It is a nutrient metal chelator. That means it binds nutrient metals. For example, manganese, magnesium, zinc, and cobalt. And these are elements that we need in trace amounts. So if glyphosate is binding these, there is a possibility that those elements are not going to be available to the animals or the humans that are going to ingest those GM crops. In addition, glyphosate is known to be a DNA damaging substance, a DNA mutagen. There is a strong suspicion now that glyphosate formulations and possibly glyphosate alone are EDCs, endocrine disrupting chemicals. In 2015, the World Health Organization's cancer agency, IARC, classified glyphosate as a probable human carcinogen. So, in other words, it probably causes cancer. Glyphosate herbicides are made of glyphosate plus adjuvants. These are added ingredients. These adjuvants simply allow the active ingredient to work better. In other words, they enhance the toxicity of the glyphosate. And the adjuvants can include chemically modified talloamines, in particular POEA. This substance is now considered so dangerous that the German government has more or less banned it. These substances, these adjuvants, are sometimes claimed to be inert, but they're not biologically inert. They do have an effect, and there are many studies showing this. They can be highly toxic in small amounts. So increasing evidence shows that some adjuvant components are more toxic than the so-called pesticide active ingredient glyphosate. Endocrine disrupting effects of glyphosate. Endocrine disruptors are, it's a particularly worrying field because endocrine disruptors can have toxic effects at extremely low exposure levels. And glyphosate and glyphosate formulations, in particular Roundup, They have tested these formulations in embryos, and they found that they caused retinoic acid interference. Now, this is something that can cause birth defects. Related to this is the reason why pregnant women are often advised not to take vitamin A supplements, because it can result in this same interference and end up with a child with birth defects. Glyphosate is known to cause estrogen interference. It mimics estrogen action. It augments estrogen function. This means that it could lead to organ damage and promote the growth of tumours. The levels detected of glyphosate to date in humans are below the regulatory safety limits, but are they safe? Nobody knows. There is a need for long-term animal toxicity feeding studies at or below the acceptable daily intake or the chronic reference dose in the US, because at the moment scientists simply don't know whether the levels detected in humans are actually making us sick or not. So the conclusions are, from this section, that there is an increasing body of scientific evidence which shows the disruptive effect of the GMO transformation process. There's clear evidence of toxicity of GM crops, especially to the liver and kidney function. And that shows up in animal feeding studies, even of a short-term 90-day nature. Cause of signs of toxicity, do they result from the GMO transgene function, the herbicide, or the mutagenic effect of the GM transformation process, or combinations of the above? Again, we still don't know. Toxicity needs to be confirmed or refuted in lifelong animal feeding studies, and these are simply not being done, and scientists are too frightened to do them. Now, this study is a study that you may have heard of. This is the controversial Seralini study, which was retracted but subsequently republished by another journal. This toxicity study was set up to extend Monsanto's 90-day feeding study on a Roundup-tolerant genetically modified corn, maize. It is the only animal feeding study looking at the long-term health effects of a complete Roundup herbicide formulation. So in many ways, this is a very pioneering study. The way the study was designed was that 20 control rats were fed non-GMO, parent variety isogenic corn. 60 rats were fed NK603 plus Roundup. This is a genetically modified corn plus Roundup. 60 rats fed NK603 GM corn alone, and 60 rats fed Roundup alone without the GM corn. Of the Roundup groups, there were 3 different doses. The top dose was half agricultural use dilution. That's a very high dose, and it's such a high dose that you and I would probably not be exposed to it unless we were trying to commit suicide, which tragically is something that happens with herbicides. The mid dose was 400 milligrams per litre. The lowest dose is the one that we're interested in. This was an extremely low dose, 50 nanograms per litre, and this is well within, it's well under the limit that is set for drinking water in the EU. So this is a perfectly legal limit for us to be exposed to. It's the kind of concentrations that you might find in contaminated tap water. So Seralini's major findings, all treatment groups showed clear toxic effects, especially to liver and kidney function, which led to increased premature death. This is the GMO and the pesticide. Males died mostly from pathologies in liver and kidneys, and there was escalation of signs of toxicity that was seen in Monsanto's 90-day feeding trial. So, in other words, what was seen in Monsanto's 90-day trial, which Monsanto said was not biologically significant and did not matter, these signs of toxicity escalated to fully blown organ damage. The females died prematurely, mostly from mammary tumours and pituitary dysfunction. the mammary tumours were statistically significant versus the controls in the lowest dose Roundup treatment group. The low dose toxicity from Roundup at levels permitted in drinking water and vastly below the acceptable daily intake in all regions can be attributed to endocrine disruption. Now, a follow-up study to the Seralini study has been carried out, I'm very pleased to say. This is a study on the female animals given the lowest dose of Roundup. That's why I mentioned to you that it was this dose that we're looking at. This dose is of interest. This was a transcriptome profile analysis of Roundup effects on the genes regulating liver and kidney function. So the activity of over 28,000 genes was measured using microarray technology. This new study was led by Dr. Michael Antonio of King's College London. It analysed the liver and kidneys from 10 female rats in the Seralini study that had received the lowest dose of Roundup in their drinking water. And these were compared with the liver and kidneys of 10 control animals who received plain drinking water with no Roundup. This lowest dose resulted in a daily intake for the rats that is 75,000 times below the EU acceptable daily intake for glyphosate and 437,500 times below the US chronic reference dose. So these are a level set by the regulators because they're said to be safe for all of us. This dose was far below the level claimed by regulators to be safe to consume on a daily basis over the long term. In other words, for the rest of our lives, according to the regulators, we could be ingesting this dose and be absolutely fine. The researchers subjected the rat's liver and kidneys to this transcriptomics analysis, and this measures the level of expression or function of all the genes present in the animal. It's an established method that's highly predictive of the health or disease status of the organ system under test. The findings were that over 4,000 genes in the liver and over 4,000 genes in the kidney were either reduced or increased in their expression in the Roundup treatment group compared with controls. The results were highly statistically significant. Over 1,000 gene functions were similarly disturbed in both organs. So here you see a kind of graphic representation of the statistical analysis of those genes. The red spheres are the Roundup treated organs. So on the left you see female livers, on the right you see the female kidneys. The red spheres are the Roundup treated rats and the green spheres are the control rats. And you see that they completely separate out quite clearly. The Roundup treatment samples are extremely separate from the controls. So this means that these effects are absolutely real, they're absolutely reliable, there is no confusion about these results. So the gene expression changes reflect the liver and kidney pathologies that were found in the Seralini study. They really confirm those results, or at least reflect those results. The alterations in the gene expression profile in both liver and kidneys correlated with disease states such as fibrosis, that means scarring, necrosis, areas of dead tissue, phospholipidosis, which is disturbed fat metabolism, and damage to mitochondria, the centers of respiration in the cells. So you see here, this chart is actually the diseases that were seen in the Seralini study on the basis of examining the organs, and this transcriptomic analysis reflects those results. Transcriptomics can't predict disease or health states with absolute certainty, and that's because not all the changes in gene function that you see in the analysis actually result in the diseases suggested. So definitive proof has to be provided by additional molecular profiling, which actually measures directly the organ's composition, and that's able to provide a direct indicator of health or disease status. And these tests, these follow-up tests, are actually underway, so they're being done right now. But the shock finding in the studies done so far, data obtained on gene functional disturbances can account for the liver and kidney pathologies, the necrosis and the failure seen at the whole organ or macroscopic levels in the Seralini study. Biomonitoring studies in humans actually suggest that many of us carry more glyphosate in our bodies than the level found to be toxic over the long term in these two studies. So Roundup at extremely low glyphosate dilutions, well below doses that regulators say are safe, can disrupt the liver and kidney gene function when consumed on a long-term basis. As a result of the concerns over the effects of glyphosate, a group of scientists actually published a paper, a consensus statement on the problems with glyphosate herbicides. And the conclusions were that human exposures to glyphosate are rising. Glyphosate is now authoritatively classified as a probable human carcinogen. And regulatory estimates of tolerable daily intakes for glyphosate, both in the US and the EU, are based on outdated science. So based on the available evidence and the inadequacy of the tests requested by regulators, at present no GMO crop and food can be claimed to be safe to consume, especially on a long-term, lifelong basis. And Cornell's Alliance for Science is most definitely misleading the public in claiming that the GMO safety debate is over. And that's all I have to say. But thank you very much for listening. I know it was a very technical presentation. It's not easy for me either. I'm not a scientist. I just communicate with scientists. So if you have any questions, I'd be delighted to answer them. And thank you for listening.